Hello everyone, welcome back. Today we will discuss one important topic of plant physiology, that is phytochrome. As we know, light is the source of energy. In addition to this, light is an important environment signal to initiate flowering or to initiate seed germination. So these processes are controlled by this pigment, which is phytochrome. So our today's learning objectives are: we will discuss about its introduction, then its discovery, historical background and structure of phytochrome, then types of phytochrome, and its mode of action. So let's start. So what is phytochrome? Phytochrome is basically a blue pigment protein that absorbs red light. So it is a basically red light receptor. As we know that light is not only the source of plant energy, it is also an important environmental signal that controls or instructs the growth and behavior of plant. So light basically control करती है growth or behavior of plant. As we know, this phytochrome is a blue protein pigment and it is responsible for the perception of light, and it controls the various photophysiological processes or initiate various photophysiological processes. And its molecular mass is about 125 kilodalton. Basically phytochrome exists in dimeric form. So इसका जो एक सब यूनिट है उसका weight होता है 125 किलो dalton, and it is the red light receptor in plant. There are some blue light receptors also in plants like Cryptochrome and phototropins. So blue light receptors in plants are Cryptochrome and phototropins. This phytochrome is the only biological pigment that is present in reversible form. So it exists in interconvertible forms. There is one form which absorbs the red light, that is commonly known as Pr form, and the other form which absorbs far red light, it is known as PFR form. So it exists in two forms, that is Pr and PFR form. And possibly this is the only photoreceptor which controls the photoperiodic responses and flowering process. So this is the only photoreceptor that controls these photoperiodic responses and flowering processes. So it exists in two forms. One is Pr form, that is inactive, and other is PFR form, which is active form. When Pr form absorb red light, then this Pr form is converted into PFR. When this PFR form absorb far red light, it is converted back to Pr form. Next comes phytochrome mediated responses. So phytochrome controls various plant responses, like it controls the seed germination. It also helps in de-etiolation of etiolated seedlings. Etiolated seedlings are the seedlings which are grown in dark conditions. So these are pale colored, longer than normal seedlings. तो उनकी de-etiolation means formation of chlorophyll in these etiolated seedlings. ये response भी control करता है phytochrome. Then it also helps in inhibition of stem and petiole elongation. Then it also helps in chloroplast development, chlorophyll biosynthesis, then leaf expansion and branching in plants. It also initiates flowering in plants, and it also controls senescence in plants. So these are some phytochrome mediated responses. So this Pr is inactive form, it is converted into PFR form in the presence of red light. Then this active form will initiate these processes. When this PFR form absorbs far red light, it is converted back to inactive form, that is Pr form. Next comes localization. So it is distributed in plant kingdom and detected in all monocots, dicots, gymnosperms, ferns, mosses and algae except fungi. So detected in different plant parts. सभी plant parts में ये detect किया गया है, and present in the nucleus and cytosol. Nucleus में भी phytochrome present होता है, and it is also present in the cytoplasm. And highest levels are found in etiolated seedlings. सबसे ज़्यादा amount है phytochrome का that is found in etiolated seedlings, and the etiolated seedlings are the seedlings which are grown in dark conditions, especially in the meristematic region. Means जो growing apices है वहाँ पे सबसे ज़्यादा amount इसका मिलता है in etiolated seedlings. So in this photograph you can see here, this is the normal grown seedling under light condition. So these are dark green in color, while this pot shows seedling with, these are the etiolated seedlings जो कि dark conditions में grow की गई हैं. सो दीज आर नोन एज इटियोलेटेड सीडलिंग्स, और इन इटियोलेटेड सीडलिंग्स के मेरिस्टेमेटिक टिश्यू में सबसे ज़्यादा अमाउंट प्रेजेंट होता है फाइटोक्रोम का। नेक्स्ट इज डिस्कवरी ऑफ फाइटोक्रोम। So its discovery is related to flowering in plants. The credit goes to Henricus and Borthwick. They observed the lettuce seed germination and observed that this pigment is responsible for seed germination in light sensitive seeds. So its discovery is related to flowering and plays important role in various plant responses which are other than photosynthesis. So ये जो pigment है ये प्लांट के बहुत सारे ऐसे रिस्पॉन्सेज में रोल प्ले करता है विच आर अदर देन फोटोसिंथेसिस, एंड दीज ऑल रिस्पॉन्सेज कम्स अंडर दिस फोटोमॉर्फोजेनेसिस. So Borthwick and Henricus in 1932 showed that red light, that is 630 to 680 nanometers, elicit or activate the germination, or we can say stimulate the germination of lettuce seeds, whereas far-red light has inhibitory effect on this process. So red light is responsible for the germination of lettuce seeds, while this far-red light is inhibitory, और जो far-red light है वो inhibit करती है seed की germination को in lettuce seeds. So these are the photographs of, this is the photograph of lettuce seed which are germinating. Then next workers were Flint and Moilister. They observed that red light promoted seed germination in lettuce seed. They also observed the same phenomenon. And Borthwick, Henricus and Parker in 1952 reported that red light inhibition of flowering in xanthium can be reversed by far-red treatment. So xanthium is a short-day plant. It is this plant's flowering is inhibited by red light, and that can be reversed by the treatment of far-red light. So अगर हम इस short day plant को far-red light देते हैं तो इसमें flowering होगी. Then Borthwick Henricus concluded that this phytochrome exists in interconvertible form, that is Pr and Pfr. So this pigment exists in interconvertible form. The next was worker were Butler et al. And Butler and co-worker, we can say, firstly isolated it from etiolated oat coleoptile. So Butler and his co-worker were the first to isolate this chemical substance from etiolated oat coleoptile and use the term phytochrome, and he was the first person to use this term phytochrome. Next was Singelman and Ferrer. They extracted this from etiolated maize leaf. They have extracted this chemical from the dark grown maize leaf. Next comes structure of phytochrome. So it exists in dimeric form. So phytochrome is a soluble chromoprotein with a molecular mass of 250 kilodalton. It occurs in a homodimer form and made up of 2 subunits. 2 subunits से मिलकर बनता है ये, और एक सबयूनिट का molecular weight होता है 125 और 125 kilodalton. Each subunit consists of 2 components. जो एक सब यूनिट है that is made up of 2 components. One is light absorbing pigment molecule जिसको कहा जाता है chromophore, and the polypeptide chain that is apoprotein. So 2 parts से मिलके बनता है एक सब यूनिट chromophore and apoprotein, which initiate this apoprotein initiates the signal transduction pathway. So ये signal transduction pathway को initiate करता है, and the apoprotein monomer has, जो single subunit है apoprotein मोनोमर का, उसका मॉलिक्यूलर मास होता है 125 kilodalton. So this is the photograph diagram which shows the structure of this phytochrome. It exists in 2 proteins, each with 2 domains. So this is the chromophore region, that is the light absorbing part of this phytochrome. Then second is the protein part which have the kinase activity, or that triggers the signal transduction or cellular responses. And so this phytochrome exists in dimeric form made up of 2 monomeric. So chromophore is a linear tetrapyrrole. जो chromophore है वो एक linear tetrapyrrole है, means it consists of 4 pyrrole rings, and it is a ring of attached to the protein through thioether linkage. और ये attach होता है protein के साथ with the help of this thioether linkage to a cysteine residue, जिसमें कि cysteine एक अमीनो एसिड है, उसके रेजिड्यू के थ्रू ये ज्वाइन करता है with the chromophore. So this is the diagram. This one is the protein part of this phytochrome, और ये जो प्रोटीन पार्ट है ये ज्वाइन होता है इस क्रोमोफोर के साथ. दिस इज द क्रोमोफोर विच कंसिस्ट ऑफ 4 pyrrole ring A, B, C and D. So pyrrole is a heterocyclic compound which contain nitrogen. और ये जो pyrrole ring है ये अटैच होता है this protein with this protein apoprotein with the help of this cysteine residue. So cysteine act as a bridge between this this protein apoprotein and this chromophore, and it is joined by this thioether linkage. The principal difference between the Pr, that is the phytochrome which absorb red light chromophore, and the PFR chromophore appears to be a cis trans isomerization. So ये जो दो forms हैं फाइटोक्रोम की एग्जिस्ट करती हैं, दो फॉर्म है PR that is cis form and trans form, and this isomerization of methane bridge between the ring C and D. So ये जो isomerization होती है ये होती है इस C और D ring की orientation change होने से. So in this photograph we can see here this is the cis form which is inactive form, and this is the trans form which is active form. So trans form is activated form while the cis form is inactivated form. So this is the difference in the chromophore of this PR form and PFR form. This is again the photograph which shows the same. So this is the cysteine residue which shows thioether linkage with this tetrapyrrole ring. This is cis form, this is trans form, and this is activated form. Next comes types. So phytochrome are of two types. Type 1, it is found in dark grown seedlings. जो etiolated seedlings हैं उनमें present होता है और इसका molecular weight होता है 124 kilodalton, and it is the most studied and times more abundant than the type 2. ये सबसे ज़्यादा स्टडी किया जाने वाला phytochrome है और ये abundance में present होता है as compared to type 2. So maximum absorption of PR is 666 nanometers and that of PFR is 730 nanometers. So ये phytochrome का जो PR फॉर्म है वो ये लाइट को absorb करता है, and this PFR form absorbs the 730 nanometers light. It has 5A gene, PH5A gene, and then next comes type 2 phytochrome. It is found in light grown plants and has molecular weight of 118 kilodalton. It is less abundant as compared to type 1. Maximum absorption of PR form is 654 nanometer while that of PFR form is 724 nanometer. It has genes, that is PHY, B, C and D and E genes. So phytochrome is encoded by multi-gene family. So जो phytochrome है ये बनाया जाता है multi-gene family से, and these genes are PHYA, B, C, D and E. So these multiple genes encodes for this phytochrome. PHYA and PHYB are the most active forms. Next comes dual form of phytochrome. So it consists of an interconvertible form, ये हम डिस्कस कर चुके हैं, known as PR, that is red light absorbing form, और PFR form, that is far red light absorbing form, and exhibit absorption spectra of PR and PFR have broad overlapping absorption spectra. इन दोनों का एक absorption spectra होता है. It means they absorb a particular wavelength of light and this spectra overlaps with each other. That PR absorb light of 660 nanometer. जो रेड लाइट PR फॉर्म है वो रेड लाइट एब्जॉर्ब करता है, दैट इज 660 नैनोमीटर, एंड इट इज कन्वर्टेड इनटू PFR फॉर्म. जब PR फॉर्म रेड लाइट को एब्जॉर्ब करता है सो इट इज कन्वर्टेड इनटू PFR फॉर्म. व्हेन दिस PFR फॉर्म एब्जॉर्ब फार रेड लाइट, दैट इज 730 nanometers light, it is converted to PR form. So it works as a light switch. जो phytochrome है light switch की तरह एक्ट करता है. PFR form is physiologically active form that we have already discussed. Red light treatment of PR results in about 85% PFR form formation and 15% PR form formation. When we treat this PR form with red light, so it is converted into 85% PFR and 15% PR form. While that far red light treatment of PFR results in formation of 97% PR form and 3% PFR form. So this is the interconversion of these two forms of phytochrome, that is PR form. जब PR form को red light provide करते हैं, so it is converted into PFR form, and this is the physiologically active form. It initiates various physiological processes in plants. When we provide this PFR form with far red light, it is converted back to PR form, which is inactive form. And this is the absorption spectra of this phytochrome. This is in the red light and this is in the dark light. So Pr form is maximum at dark period, and this red form is this PFR form is maximum at red lights when we provide it this pigment with red light. Next is difference between PR and PFR form. So firstly, PR form, it is blue green in color and it is light green in color. This PFR form is light green in color. Then PR form, as we know, this is inactive form of phytochrome, while this is active form of phytochrome, and this form mediates the various responses. So PFR form is the activated form. In this PR form, it shows the maximum absorption spectra at the region of red light, that is 680 nanometer, while this PFR forms the maximum absorption spectra at far red region, that is 730 nanometer. And this PR form can be converted into PFR form when we provide it with red light, while this PFR form can be converted to PR form when we provide it with far red light. So it is found in diffuse throughout the cytosol. ये cytosol में ज़्यादा होता है, and it is found in discrete areas of cytosol. Particular कोई restricted areas में PFR form present होता है cytosol के. Next difference is PR form contains many double bonds in pyrrole rings. जो pyrrole ring है, tetrapyrrole ring, उनमें बहुत सारे डबल बॉन्ड्स प्रेजेंट होते हैं PFR फॉर्म में, while in PFR form it contains the rearranged double bonds in all the pyrrole rings. इसमें rearrange हो जाते हैं double bonds. So there are some differences between PR form and PFR form. Next comes actions. So the amount of light is referred to as fluence, that is the number of photon hitting on a unit surface area. So किसी भी प्लांट के ऊपर जो अमाउंट लाइट की पड़ती है उसको फ्लुएंस कहा जाता है, and fluence is the number of photons hitting on a particular unit surface area of the plant body. So each phytochrome response has a characteristic range of light fluence. So जो phytochrome response है वो mediate होता है fluence से, that is how many photons are hitting on a particular portion of the plant body, over which the magnitude of response, जो magnitude है और effector response का, is proportional to the fluence. So it is directly controlled by this fluence. Means कितनी light किस particular area में पड़ रही है, so that will initiate the response, light mediated response. So these responses can be categorized into three major groups. तीन groups में हम इसको divide कर सकते हैं on the basis of their sensitivities to fluence or light. They are namely very low fluence responses, abbreviated as VLFR. Second is low fluence responses, जिसको abbreviate किया जाता है LFR, and finally the high irradiation responses. So firstly, very low fluence responses, abbreviated as VLFRs. So these responses are sensitive to very low level of light stimulus or fluence. So these responses are extremely sensitive to low levels of light and fluence, and it requires light fluence of 0.1 nanomoles per meter square. So this is the range of this very low fluence response. So small exposure of light is needed to induce this VLFR response and convert less than 0.02% of total phytochrome to PFR form. So only a small amount of this PR form is converted to PFR form, that is 0.02%. Therefore, the far red light cannot reverse VLFRs and this very low fluence responses, and they are non-photoreversible responses. ये जो responses होते हैं इनको हम far red light provide करके reverse नहीं कर सकते हैं. So that's why these are known as non-photoreversible responses, and very low proportion of PFR is effective for these physiological events. और ये जो भी events हैं इसके लिए बहुत ही small amount की requirement होती है PFR form की. For example, that can be provided by the Arabidopsis seeds. So ये जो Arabidopsis की seeds हैं, this Arabidopsis is the member of family Brassicaceae, and their seeds, this Arabidopsis seeds can germinate with very low red light requirement. सो ये जो सीड्स हैं इनको जर्मिनेशन के लिए बहुत ही कम क्वांटिटी की लाइट की ज़रूरत पड़ती है. सो दिस ग्राफ शोज दिस VLFR response. So this is reciprocity and not far FR reversible. जो ये responses हैं इनको हम far light provide करके reverse back नहीं कर सकते. Next comes low fluence responses. So such responses include classic red and far red photoreversible responses. So ये जो responses हैं ये red और far red photoreversible responses हैं. Here low fluence responses 1.0 micromole per meter square to 1000 micromole per meter square. So this is the amount of light required to induce this fluence or response. It includes most red and far red photoreversible responses like promotion of lettuce seed germination and leaf movement. So this low fluence response can be reversed. So it is interconvertible. It can be reversed with FR light, that is far red light. And last one is high irradiation responses that is abbreviated as HIRs. So some photomorphogenetic responses require prolonged or continuous exposure of light of high irradiance and are proportional to irradiance. So कुछ एक ऐसे प्लांट के responses होते हैं जिनको बहुत ज़्यादा prolonged requirement रहती है light exposure की, and after getting this light exposure, prolonged light exposure, they can induce these photomorphogenic responses. So these responses are known as HIR, that is high irradiation responses, and are not photoreversible. और ये जो responses हैं इनको photo reverse नहीं किया जा सकता. Examples of HIRs are anthocyanin synthesis in dicot seedling and apple skin. So जो anthocyanin accumulation होती है dicot की seedling में और जो apple की skin है उसमें जो red color होता है that is due to accumulation of anthocyanin. So this is due to prolonged exposure to light, and this response cannot be reversed. Next HIR is inhibition of seedling elongation, that is inhibition of hypocotyl growth, and flower induction. So this cannot be reversed. Next is plumular hook opening, cotyledon expansion in mustard, and ethylene production in sorghum. Then de-etylation of seedlings. However, the effect is not photoreversible. जो ये सारे के सारे effects हैं इनको photoreverse नहीं किया जा सकता. So these are some responses which comes under these HIRs. So this is a graph which shows prolonged exposure to this light, and only then the plant is able to show the photomorphogenetic responses, and these responses cannot be reversed by providing far red light. Next comes functions of phytochrome. So it regulates the germination of seeds, and the term used for this is photoblasty, synthesis of chlorophyll, then elongation of seedlings, size, shape and number and movement of leaves, and the timing of flowering in adult plants. So this is also controlled by this phytochrome. This pigment has a regulatory role in all phases of plant growth and development, that is commonly known as photomorphogenesis, and is apparently ubiquitous in all taxonomic group of eukaryotic plants with the exception of fungi. So सभी के सभी plant groups में ये अपने responses show करता है except fungi. Next is photomorphogenesis. The process by which plant development is controlled by light is known as photomorphogenesis. So when light controls the development of plant, that process is known as photomorphogenesis. Plant exhibit different growth and habits in dark and light. In dark, they have elongated stem, undifferentiated chloroplast, and unexpanded leaves. So these plants are commonly known as etiolated plants. And this is called scotomorphogenesis. So seedling which are plants which are grown under dark conditions, and these organ formation, this process is known as scotomorphogenesis. So light grown involves the inhibition of stem elongation. When the seedlings plants are grown in light, so there is inhibition in stem elongation, then there is a differentiation of chloroplast, means formation of chloroplast, and accumulation of chlorophyll and expansion of leaves. So this is just reversal of dark grown seedlings. Thus the same stimulus causes opposite effects on the cell elongation and in leaves and stem. Photomorphogenesis can be induced by red, far red and blue light. And this pigment absorb light and can be altered electronically. जैसे ही pigment light absorb करता है, so it becomes activated or inactivated. So it results in change in other chemical in the immediate environment to drive photosynthesis. So it also helps in changing the chemical composition of other chemicals and various plant processes including photosynthesis. So this is the seedling which is grown under dark condition. इसमें जो stem है that is elongated, leaves expanded नहीं है, while this is the dark grown seedling जिसमें stem की growth inhibit हो जाती है and leaves expanded है and they are green and chlorophyll is differentiated. So this is the blue light, this is red light. So blue light receptor in the cell is cryptochrome, while red light receptor is phytochrome. So these receptors helps in differentiation of these chloroplast. These are the etioplast which are found in dark grown seedlings. जो etioplast है ये dark grown seedlings में होता है. When they come in contact with this light, so they are differentiated into the chloroplast which are green in color. Then this process is known as photomorphogenesis. The next is seed germination. This process is also controlled by this phytochrome. So seed germination is inhibited by far red light and is stimulated by red light. Seeds deposit under heavy canopy of trees don't germinate. जो plants, जो seeds heavy canopy means dense forest में होते हैं, उनमें जो seeds हैं they are unable to germinate because they didn't get enough exposure of light. Some seeds germinate only in spring because they become exposed to red light after the leaves have fallen and decomposed and before the new leaves that shade them have merged. So कुछ एक plants के जो seeds हैं वो germinate करते हैं spring में because जो fallen leaves होते हैं वो decompose जाते हैं spring में and the new leaves start forming and वो shade provide नहीं करते हैं germinating seed को, only then these seeds can germinate. The phytochrome protein is generally synthesized in the Pr form when red light enters to the plant, and some Pr form is converted to PFR form. In some plants, PFR form enters the nucleus and binds to the protein that are associated with the DNA either to activate or inhibit the transcription of specific gene. So जो PFR form है ये enter करता है nucleus में और वहाँ पे it binds with some specific proteins on the DNA and it acts as a switch. It can activate or inhibit the transcription of certain required genes. So this is the phytochrome, this is the PR form and it is converted to PFR form which is activated form, and this form is responsible for seed germination. Then there are some MCQs on this photoperiodism and phytochromes that you can solve and give its answer in the comment section. So this is all about this today's discussion. If you have any questions, queries, any suggestions, you can give it in the comment section. Thank you. Have a great day.